From Project ALS, I'm Valerie Estess,
And I'm Erin Fleming, and this is From Lab to Life,
a podcast where ALS research leaves the laboratory and connects with real lives.
and where the doctors and scientists working to unlock the mysteries of this fatal brain disease sit down with those with ALS and have honest conversations.
Bridging this gap is important. It helps ALS research not feel so far away for those with ALS.
Because when you bring the research out of the lab, it impacts real lives and pushes the needle even further to finding a cure for this disease.
Today we meet three remarkable people who are, well, in a relationship. Over the last year and a half, that relationship has intensified. When Melissa Diaz Viera, a social worker, wife, and young mother, was diagnosed out of nowhere with ALS, she had questions. Where was the medicine? What was Project ALS doing to figure out ALS, to slow it down and cure it? Well, one of the efforts that Project ALS is most excited about was unfolding in the laboratory of Stephen Altschuler and Lani Wu at the University of California, San Francisco. Mathematicians by training, Steve and Lani had moved through Harvard and Microsoft, pursuing their passion to discover new cancer drugs through deep learning. It was Project ALS mentor Tom Maniotis who suggested that Steve and Lani might do for ALS what they were doing for cancer. Turns out Steve and Lani had a lot to learn from Melissa, and Melissa from them, Erin.
That's very true. And I think from the outside, the topic that we're discussing today seems particularly like basic science, something that's really intractable and not understandable to someone who isn't a machine learning expert. But the reality is that any of us confronting ALS, either through research or lived reality, immediately hears the word heterogeneous when we talk about this disease. We're told that despite ALS being a so-called rare disease, no two cases are alike. There's no way to predict how a single case of ALS will progress or to identify subtypes that could lead to more precise diagnosis, clinical care, and ultimately therapeutic intervention. And in this sense, we're in the ALS world really lost at sea. So the question that this group today is asking, what if we could find islands in that sea of ALS and anchor subtypes of ALS to these islands in a way that's objective, predictive, and reproducible? And I think today's conversation is between three people who are working together to do that. So on that note, Melissa, I'd like to ask you the first question. From your vantage, living with ALS, working at an ALS research nonprofit, what do you personally find engaging about the seemingly very basic research project that Lani and Steve are leading at the Project ALS Therapeutics core?
I think that when I was first diagnosed, all of this was so foreign to me. And it was really hard to wrap my head around all of the research. It's such a complex disease. So when I met Steve and Lani last year at our therapeutics core retreat, I really loved how clear their research was. There seemed to be such a straight line from where they were starting to where they're hoping to go. Thinking about being able to collect a sample from a live patient, creating something out of that to be able to stratify and create categories for these patients, and then being able to hopefully utilize treatments that are already created or could be easily created to then be able to use that in patients. It's just a very clear path. And I think for a lot of patients, we don't see that clear path sometimes. Some things feel very exploratory. They feel like we're still just trying to figure out this disease. So that really stood out to me. It gave me hope. It made me feel like maybe certain treatments may not be as far away as we, usually see. And so that really helped. It also stood out to me how much Steve and Lani really wanted to connect with patients and use patient experiences to also inform their work. And I think that is something that is missing sometimes. And so that's been a lot of my focus, is trying to create more of a connection between patients and researchers so that we can help each other and move some of this work forward.
I don't want to take over the lines of questioning here, but I will ask Lonnie and Steve something, and then please feel free to to take it on yourself going forward. But Valerie mentioned that you came to ALS, rather late through Tom Maniatis at Columbia, and that you had worked in a lot of other fields before you made it here. Could you talk a little bit about what made you interested in ALS and what your experience applying knowledge, technology, and research, that had other very diverse ends before, what that's looked like as you've been now in ALS for almost 10 years, right?
That's right. Well, I guess for us, the two most stark challenges that were in contrast to what we were used to seeing in cancer was first that everyone kept telling us the disease was heterogeneous, but we didn't really see a way forward. The genetics, unlike in cancer, are just so unclear for ALS. And the second challenge was what kind of material can you look at as scientists to try to understand how to get to some place where you can try to bring some sort of order to all the chaos. So in cancer, what has really helped with both understanding the disease and bringing forward therapeutics is the ability to stratify based on genetics. But where do you get those genetics? You get them from tumors, which patients are very happy to get rid of and have analyzed. With ALS, that heterogeneity is hard to look at genetically, and it's very hard to get samples, that are disease relevant. So those two challenges, I think that was very inspiring to try to figure out a way through that.
I think we struggle with this project for a long time because the individual differences. And we were, for a long time in the very beginning, we were really just identifying individual differences, not the disease differences. And then another thing is that the rarity of ALS causes us to have a small number of samples. How can we really detect signal from a small number of samples and within the heterogeneous disease scope? It was really hard for us to figure out, did we really get something real or not?
So just to be clear, for people who don't know, ALS is heterogeneous, meaning no two cases are alike. But about 10% of ALS cases are familial, are inherited, and do have genetic mutations associated with their progression. But Melissa, you are a so-called sporadic patient. So the genetic underpinnings of your disease is, unknown. So it was really interesting to hear that you saw a straight line between what Lonnie and Steve were doing in their lab to information that was helpful to you. What was it about that? I mean, minus genetic addresses or genetic targets, you have sporadic ALS. We don't know the genes that are driving your disease per se. So why was their work of straight line interest to you? Just curious.
I think that the approach of trying to find sporadic cases that are similar, have similar characteristics to certain genetic cases, allows for the possibility of utilizing gene therapies for sporadic cases. And we know that right now, gene therapies seem to be kind of the most promising, that we're seeing some results in patients right now. And so that definitely does give hope to people with sporadic cases. And it's the closest thing that we have, that we could potentially have right now, to actually having a treatment that is a little bit more individualized rather than trying to have a one size fits all type of treatment.
Right.
I've not seen a lot of success with that because it is heterogeneous. And so even though, ALS patients share a lot of qualities, and yes, we know that there are protein aggregates and neuroinflammation and lots of different factors, there still hasn't been one treatment that can help all patients with all of these different types of characteristics. So this felt like we could get a little bit closer to a bit more individualized and more data based for each specific individual.
Right. So I'm just wondering, if Lonnie and Steve, if you could sort of just outline for newcomers your platform. How are we now discovering that there may be differences, but also similarities among people who have familial ALS and people who have sporadic ALS? How is that even possible? But can you just describe your screening platform and how mathematicians go about creating a platform for such discovery?
I'm not sure I consider myself a mathematician anymore, but maybe people with mathematical training and ways of thinking. So when we started to understand the disease from reading and talking to people, we sort of got this picture in our head that there was this, as Erin said, this big ocean with tiny little islands, and those tiny little islands are the genetics we know. And as you said, 90% are called sporadic, 10% are familial. Of that 10%, we've identified about half of that population's genetics. So that leaves 95%, this blue ocean. So, to try to get at the question of how can we, find more islands out there and start charting that ocean, we had to make really two assumptions. One was that, and we had no idea if this would be true or not. One was that ALS, even though it's heterogeneous, both from a clinical perspective and from a molecular perspective, it's not infinitely complex. In other words, it's not the case that every person who is living with ALS is completely different from every other person. So the first is a simplifying assumption that there would be groupings. And the second, which goes back to this problem of what are you going to study as a basic scientist, was that we could find tissues to work with that would be revealing of the disease. And what we need to be able to do is to get at what Lonnie said earlier, which is that whenever you're studying a disease and you want to get at it by comparing it to something like a person who doesn't have that disease, what you want is that the signal from one person to another is smaller than the disease to healthy. So if everybody is unique, then the only signal you're going to get is who I am. But if there is something about the disease and something about the tissues you're going to study that's revealing of the disease, then what you're trying to do is lock onto the signal that's bigger than the differences between people. So the way we went at this was we had to figure out what kind of cells we could work with. And because we want to work with people who are alive and we want to help them, that means we need a rapid sort of way to look at a person and say, what kind of ALS do you have? And I'd like to say that we were really smart in choosing what we did, but we weren't. We were just lucky. So when we came to Columbia, when Tom Maniatis invited us out, we learned about ALS during that meeting. And at the end, we said, great, we'd love to help. So what kind of tissues do you have for us to go analyze? And there was this awkward moment where everyone said, well, we have a bunch of fibroblasts, frozen down. And I remember all of us thinking, how can we get at this from looking at skin cells? And we didn't want to go through the process of converting them to iPSCs to stem cells because that erases aging. And then we certainly didn't want to go through that process and then convert them to neurons because that can take many months. So we decided maybe there's a signal in the skin cells. And that's where we began. In many cases, even in cancer, you'll take one example of cancer and one example of not cancer, and then you'll just study that. But because ALS is so heterogeneous, we decided to sort of bite the bullet and not just pick one sample of ALS and compare it to one sample of healthy, but rather we decided the more samples we could get, the better. So together, our platform is looking at lots of samples from people who are living with ALS and not, and trying to find a signal in skin cells.
Prior to this, Steve and Lonnie, had anyone looked at skin samples for ALS clues?
I believe there has been work in the past, not just in ALS. Actually, in the past 5 years, there was work at Google looking at Parkinson's fibroblasts, where they tried to find a signal between Parkinson's and not. I think there is a history, maybe even Alzheimer's, looking at skin cells. But for our purposes, to try to stratify, we need to really dive into a collection of ALS fibroblasts and age-matched non-ALS.
Melissa, I'm wondering, listening to Steve describe this platform, and of course, we've been up close with this for a while, but I'm wondering if you could talk a little bit about what your experience as a so-called sporadic ALS patient has been, what that looks like in terms of, what your clinical path has looked like, and maybe, what does being able to identify an island or map yourself onto an island, what would that potentially mean for you, as you think about your own experience?
Yeah, I mean, like I mentioned earlier, I think that as a sporadic case, not knowing exactly what is causing your disease, not having great biomarkers to kind of see what's happening in your specific body, you're left to sort of just try whatever, And so, in my particular case, I've tried the FDA drugs, one of which was taken off the market. I tried various supplements. I've been in a clinical trial. I continue to progress. So I'm I'm now 3 years, almost to the week of symptom onset and about 2.5 years out from diagnosis. And I am, wheelchair bound. I've lost most of my function of my limbs. I've my breathing, as you see, I'm using a vent throughout this conversation. So my breathing has been affected. And I've just very recently started having early symptoms of like swallowing and speech issues. But, you know, it keeps going. And so it's hard to feel like you don't really know what to try next and how to find something that works for you specifically. And so again, I think looking at research that tries to create these groupings, whether it's tying it to characteristics similar to certain genetic types or groupings based on certain, factors like we mentioned before, neuroinflammation, mitochondrial dysfunction, things like that. Just getting more information about yourself so that you know which therapies may have a better chance at working, I think would be very powerful.
I should say that once we start analyzing these skin samples, the goal was to try to group them together, not using genetics only because we only have that as a guide for a small number, but actually looking at how they look. So that's sort of what's called a phenotypic characterization. Because what else do you have if you don't have genetics? So the program that Lonnie and I have been working on is to try to use genetics as a guide. Take those 5% islands, then ask, can we find people who are sporadic, whose skin cells at least look like those genetic ones? And there is really hope for SOD1 that one of those islands has an FDA-approved antisense oligonucleotide. FUS is looking very promising and is in phase 3 clinical trials. So our very first project is to try to find people who are living with ALS who phenotypically, from our profiling, look like either SOD1 or FUS. So that is the direct path that I think Melissa was mentioning. And if we can find people, and there's no reason why there should be, but some of our early data suggested we might be able to find such people, then to work with the clinicians and with those companies and see if we can get compassionate use.
One question for people who listen to this story here may be asking, why are we helping with Melissa? And the reason right now is that because this is still a hypothesis, we're testing many things. For example, this require IRB a protocol. So far, we only have the IRB in UCSF. We can only look at biopsies from patients who are in UCSF. We cannot get it from Melissa's samples. We are moving forward with NGH to see whether they can uh, have protocols set up so that we can also get their samples. So I'm going to say that even though we know Melissa, we are handcuffed. We cannot analyze her biopsies and try to see whether there's a possibility. And also, this is really at a research level here right now. It's now proven that we can really apply it to all patients.
We've had people reach out and say, I have a friend or a family member, and nothing would make us happier than being able to have them, I don't know, go see a dermatologist and send us a skin sample. But
Right. That should be the easiest thing in the world, right? Yet, there are so many regulatory stipulations and limitations and challenges, but we'll get there, I think. Just I'm wondering, so Melissa, we're still at what you would, I think, characterize as an exploratory stage. I think we've kind of proved principle with Steve and Lonnie's platform. What gives you hope in their approach?
They're so focused and they're so driven and they're so patient focused. They really have the patient in mind first and foremost. Um, and I think that that's really key. I think they're also, expanding outside of their lab and working hand in hand with a clinician and meeting patients in person. I think that will expand their work as well, to give them more information that could support some of these different categories that they're trying to identify. So it definitely gives me hope. I cannot benefit from their work currently, but I'm hoping that, as their work continues, that, we'll be able to see that expand to other clinics. And, that is always a great thing to see.
Maybe I'll ask the inverse question, but a little bit more pointedly. So, Lani, you just mentioned that, of course, this is early stage research. There are limitations to who can actually provide skin samples right now. And Melissa, unfortunately, can't provide skin samples to this IRB approved project at this point. So, how has Melissa informed this research as you've worked with her over the last several years at Project ALS?
I think the most important thing is that if we are successful, we can help people. Maybe not everyone, but some of them. And they are desperately needing a help.
one of the most nerve-wracking moments of all the last decade for us was presenting last year in front of Melissa. Until then, it had been cells in a dish and reading papers and talking to you. It was all very sort of theoretical. And, before the talk, Lonnie and I said, boy, this is really important to talk to someone living with this and to see what they think. I remember we went up and we spoke, and I looked over at Melissa and her husband during the talk, and thought to myself, who am I to be here talking about this research when there's someone out there really living with this? I hope at least I've been clear in what we're trying to do. And after the talk, we walked up to her. We were so nervous, and she was so gracious. And I mean, you ask how does this make a difference? It just inspired us talking to her, and it inspired the people in our lab. And over the last year, we've been working with Dr. Kathy Loman-Hurth, who leads the ALS Center here at UCSF. There are other components to how we want to stratify, including giving digital wearables, and ultimately, where AI probably could be helpful is looking over patient clinical records. But we've had now many people in our lab go to clinic every 2 weeks since we've begun getting skin biopsies and handing out the watches to meet with the patients and to see with our own eyes the heterogeneity. Our first day, there was a patient who came in walking just fine, but couldn't speak clearly. And the very next person was in a wheelchair, could speak very clearly. These differences are just so dramatic, especially to someone who's not a clinician. And I think that has been very helpful. We also are trying to get those skin sample to fibroblast turnaround times as fast as we can. And I think, again, meeting with patients is just very inspiring. It's not just, let's go collect some cells, and sometime in the next 4 years, we'll write a PhD thesis on it. I mean, there's a sense of urgency in every person in the lab.
You don't seem out to write papers. You don't strike me as those kinds of researchers.
You write papers, but that
No, I mean, great papers, by the way, but that's not your top priority, it seems. You really want to help people who are living with disease now. But Melissa, I have another question for you. It's like, so Steve and Lonnie, you aren't clinicians, so you don't see patients. You research the disease. You work in a laboratory. But I wanted to ask you, Melissa, give me a percentage. How many of the clinical people you see, doctors, neurologists, are aware of Steve and Lonnie's work? It seems that a lot of the exciting studies across ALS research are happening in a vacuum, and most doctors who actually seeing patients don't even know that it's happening. Am I overstating this, Melissa?
Well, I think my specific doctors are very involved in research. So I think it's a little bit different. But in speaking to patients, across the country, I'm very connected with a lot of ALS patients. And you do hear a lot of patients that are coming in and they are the ones, asking the doctors questions. They're the ones trying to inform the doctors of research that is happening. Even when I was first diagnosed, I was seen by a different doctor, not my current team. And it wasn't a research hospital. And they were really not informed about clinical trials or, some of the things that were happening in the research landscape. So I think that is a very big disconnect in general. I just tried to connect myself with people that were interested in the research because I was interested in the research. But I wouldn't say that that is the norm, let's say, for all patients.
Do you think that it would help if it were the norm? That's a question for all three of you. Preclinical research. So drug candidates get to the clinical trial stage, and then it's arguable, whether or not that information is easy to access for people with the disease. But there's a level of research that happens before clinical trial. It's at a preclinical level, the level of, just discovery or proof of principle. That's what we do at Project ALS. But do you think if more people at a clinical level and people who are living with ALS knew about that preclinical research, it would empower them, it would provide confidence th- throughout the community that there are efforts being made to drive better therapies and cures? Again, the probably a rhetorical question, but I feel that there's a big gap, between what you, Steve and Lonnie, are doing and what a clinician who might be working in a clinic would know and be able to share with a patient.
Yeah, I mean, I think that there is a very big disconnect, like I mentioned earlier, between patients and researchers kind of via the clinical teams, right, as well. But um, I think that's what really stood out with me with Steve and Lonnie as well. They were able to communicate their research in a very accessible way. I think a lot of patients are they're very intimidated by research. It's this language that is, it might as well be, I don't know, some other language you don't know, right? And so it takes a lot of time to try and understand all of these terms. And it puts a lot of people off. They're like, oh, that's not for me. I don't get it. Call me when you have something that is going to cure me, right? So I think that, it is important for the researchers to try and make their research more accessible and understandable to patients because that also creates more engagement that ties into fundraising and donations and all these things that we very much need as well for this research to move forward. So I think that there is a lot of work to be done in that realm. And I think with clinics, it is tricky. Like I wish that there was more education and more conversations related to research and different options. Like I said, some clinics do a much better job at that than others. But every patient is also different. Some patients feel very overwhelmed with like an overflow of information. They're coming into clinics with lots of needs. We're only seen every 3 to 6 months typically. And in that time, there can be a lot of changes. You have to be seen by a lot of different specialists. I would say most people are in clinics like at least 4 hours typically. So it's long days. And so, all of those factors play a role. But I think doing things like this where you can try and inform the community about different projects going on can give a lot of hope. And I do think patients are very interested. They just don't always know how to access. And on the flip side, it sounds like from what Steve said, there's some of that from the researcher side and in talking to patients. There's this, it's like, am I going to say the right thing, or is my work going to resonate with them? Right. So I think all of that plays a role.
I want to get into a little insider information here. Not so exciting, but I will say Project ALS has been working with Lonnie and Steve since 2016, and over that time, they have consistently declined our requests to speak about their research. It's not a favored option for them. And I think Melissa, the what you just shared and really your interactions together. I mean, maybe I'll turn this back over as a question, Steve and Lonnie. But sort of what changed your mind or what convinced you that maybe it was time to talk about this? And to the points that Melissa just made, how is this project trying to address some of these barriers between people living with ALS and the people working on it, siloed away in their labs, never showing their faces?
Well, the reason why we agreed is because of you and because you said just what you just said. You helped us to understand how important it is. I think two things really are important to Lani and me, that we never overstate our science because we don't ever want to give false hope. And the other is, we are scientists, particularly trained in mathematics. We want to be very precise about what we claim we can do and what we can't do. And we spend a lot of time on those papers. We do write, Valerie. We write very carefully to be exact in what we've done. And I'm always afraid that if we speak publicly, we may be misunderstood. But I think the reasons for speaking publicly, as you and Valerie have been very convincing about, is that people do need to hear about research. Science needs to be heard. It needs to be put in terms that people can relate to. That's my perspective.
And then the other thing is that I think after the years are working, we now have a signal we cannot believe in on the research grade. To really move this forward, we need patients' help. We cannot do it all by ourselves. And patients need to know that this is research. And this research, I don't think that this is a project that we can test it in the lab anymore. We cannot find the right minds to do it because that even regardless what the results are, we don't know what the true impact in patients. And we need to work with patients side by side.
And with the people who are making these therapeutics,
Yeah.
which we are now doing. We're engaging with them also.
Then one thing to get back to what Melissa was talking about research. I think that the traditional research had two parts that may be relevant to patients. But one of the more prominent part is drug treatment. Steve and our program today, yes, we can get to treatment, but probably later. The first step we have is how to repurpose the existing compounds. And that is re-identify the right patient subgroup. And that is not as common people are thinking about when you coming to ask your clinician, what drug can I use? They're not thinking about, am I in this subgroup or not? This is a different type of question that most patients are thinking about. So this angle is a little bit different from most of the patients what they're thinking about just FDA approved drugs.
Yeah, and I think for me personally, that strategy has become so much more important because, as Melissa, you said at the beginning, there's sort of a direct line now. In recent years, we've seen the first hints of truly effective drugs. I mean, not cures for ALS, but truly effective drugs in these genetic subpopulations, obviously proven with tofersen in SOG1 and really promising data plus subpopulation. And so we no longer have the expectation that an effective drug in ALS should do a little bit. We know that if a drug is in the person, it can do potentially a lot. And so the sort of most urgent and imminent task in a lot of ways is figuring out how we can do a lot in more people with the available tools, while of course continuing to develop other treatments which will be needed for other subtypes of ALS. And hopefully in lockstep with drug development, this platform can keep a pace so that we don't have these very limited islands, but we can immediately or very rapidly apply effective treatments to the right people. Yes, yeah.
Well, maybe I could say something. The work that went into identifying SOD1 and FUS, it was really deep biology. And the work that, went into development of the therapeutics and the clinical trials and the people who did this work, both in the research side, the clinical side, and in the company sides, this is really profound work. It was very deep scientifically. It was, financially risky. So in fact, we should be very clear and acknowledge that anything that we're able to do in terms of identifying these populations and repurposing these drugs stands on the shoulders of really giant work and giants.
I agree with that. I was really struck by you not wanting to overpromise, which I really appreciate. But Steve and Lonnie, what is your kind of forward-looking statement here? What do you hope to achieve in the next year? And what would you say to Melissa directly about what she might feel hopeful about? What should we be hopeful about?
I think if we can identify even just one, plus one, or one, someone like patients who do not have the mutation, but they can benefit from the fast ASO or SAR1 ASO, then we are in a new era of treatment for neurodegeneration. Even one.
That is our 1 year goal.
Wow. That's a great goal. And just to be clear, to pull back for people who are new to ALS research and this conversation, the idea here is to use, and pardon me for reducing your work to AI, but using an AI platform to identify potential similarities among people who have a genetic mutation form of ALS and people with so-called sporadic ALS. But these patients may share more in common than we ever realized, thanks to the platform created by Lonnie and Steve and shared and mentored by Melissa. Tell me if I got that wrong.
No, you got that right, Valerie.
Thank you, Stephen.
But, because this is all uncharted territory, the way that we try to define what similar is, we're doing it through microscopy. We're also doing it through transcriptional profiling. And, there may be other ways to do that. And in terms of just one point about I would say that, because this is a rare disease, the number of samples is very small. I wouldn't say I would call what we're doing AI on this part. I would just call it sort of traditional, maybe machine learning, traditional analysis.
Sorry to have put you in that box.
No, the difference would be $100 million in funding. So if you want to call it AI, please feel free.
This platform is not just built by Stephen and me. This platform is built by a very talented, very devoted team in the lab. And without them, we could not have done this.
This platform is built off of a whole team that, it started funding very much thanks to Project ALS. After our visit there, we came back and we received a letter that no researcher, I believe, has ever received, which is a letter saying, congratulations, we're going to fund you. Please send in your proposal.
Yeah, we aggressively pursued you, to say the least.
That will stick with me forever. So the platform really is in our lab biologists and people with backgrounds in machine learning and AI working side by side. So right now, it really is the process of taking those samples right from the clinic, being able to grow out the fibroblasts, being able to do very careful cell culture work. We now can grow 100 to 200 simultaneous human samples and compare them. That's with imaging, also the profiling using omics technology. That goes through, also traditional biologists who are searching for what are the right biomarkers, then off to the people who are trained in statistics and machine learning, who, are finding the signal and trying to interpret it back with the biologists and the team. We will always be grateful to Dr. Neil Schneider at Columbia for providing us with so many skin samples at that time and throughout this project. Really, without him, this project would not have been possible.
No, that's great. That's great to know. We love working with you guys. And Melissa, do you feel intimidated when in your first relationship, your first exposure to Steve and Lonnie's research, and how has it changed for you? You talked about being slightly, distanced by the language of science and stuff. What have you come to understand about their research that gives you some hope?
No, they are not intimidating people. They are wonderful, warm, welcoming people, open people. So I appreciate them being so open. And I think in our conversations, we both asked a lot of questions that we really wanted to know. And I think that just opened up a lot of opportunities for learning more. And so I really appreciate that. But in January, it'll be 2 years since I started with Project ALS. And I've learned the language a lot more, I would say. I'm not an expert, not quite fluent yet, but I think that in my journey, it has been helpful to just try and understand what is happening or what could be happening, let's say, in my body. What could be some pathways forward. And, every day I have to live with the reality of the disease and see that I continue to progress, and that's really difficult. But at the same time, I get to see how many people are working really hard to figure this out and to provide some treatment opportunities for patients. And so that gives me hope. And I'm hoping that, we will be able to see those benefits. And
If I could ask one more question, Melissa, given your own experience learning a language, what would you say to other people in your position, to someone else who wants to learn more about ALS research, who is dealing with ALS either themselves or through a loved one and maybe doesn't know where to start in trying to engage in this, opaque world of research, of papers?
there are definitely publications that maybe are make things a little bit more accessible in terms of the language. I think if you're starting out with a research paper that is not created in lay terms, that is going to be more difficult. I think, watching webinars where people are explaining the research in more tangible terms, I think is helpful. That's kind of how I started. I would watch webinars like every week, and there are, different organizations that offer them. And, sometimes I would just sit down and write an email to somebody and just see if they would respond and just ask questions. I think people will surprise you. A lot of people are like, feel honored almost that somebody wants to know about their work. And so I think people are more open than we might give them credit for. In terms of research, if you want to contribute to research, there's so many different ways, whether it's through, obviously people think about clinical trials, but if you're not in a position to join a clinical trial, there are surveys, there are so many different ways to provide important information about your particular case that could help researchers use that data to inform their work. So there are lots of different ways to be involved.
And I think on that note, if anyone has questions about the topics we've discussed today directed at any of our guests, please email us. The email is info, I-N-F-O, @projectals.org. You will get a response and we will be honored. And we can direct the questions to our guests if there's something that either Lani, Steve, or Melissa would be most adept at responding to.
Yeah, I would thank you all for sharing your relationship with us, giving us a window onto it. From lab to life is the way to go. And you guys are showing us how you're leading the way. So thanks for joining us today. Any final comments?
Thank you for everything you're doing. And, it's been a joy to get to know all of you, and can't wait to see where this work goes.
Yeah, I mean, we have a big year ahead of us, according to Steve and Lonnie. And as far as I'm concerned, it's going to be a banner year. So thanks a lot, everybody. And we'll see you next time. Thanks for listening to this episode of From Lab to Life, a Project ALS podcast,
where science meets hope and every discovery brings us closer to a cure.
Be sure to subscribe to us wherever you listen to your podcasts and join us next time as we keep bringing the research out of the lab and into life.